Today we're going to continue to go over parts of the cell. Okay? We're going to talk about the eukaryotic cell. And we're going to talk about some functions of that, some organelles of the eukaryotic cell, including vacuoles. We're going to talk about the nucleus and so forth. Okay? What do we know about the nucleus of eukaryotic cells?
Sorry.
Bless you. What do we know about the nucleus of eukaryotic cells? Surrounded by a membrane. That membrane is, how would you say that membrane is? Describe that membrane.
Pores.
It has pores. It's a nuclear envelope. Is it what? Is it? What do you think it's made of? Probably phospholipids. Is it a single layer membrane? Double layer membrane. All right, with proteins and pores as well. Okay. What's held inside of that membrane? Genetic material, which we know is DNA. Okay? So it says the nucleus contains most of the DNA in the eukaryotic cell. Why does it say most and not all? Well, DNA. We're talking about DNA. So the question basically is then, I could reword it by saying, where else in the cell will you find DNA? Where else outside of the nucleus will you find DNA? Where in the cytoplasm? Not ribosomes. The mitochondria and the chloroplast. Okay? Because they actually are derived from bacteria, mitochondria and chloroplasts have their own DNA. All right? But most of the DNA, the bulk of it, is found in the nucleus. All right? Ribosomes use the information from DNA to make protein. So we go through the nucleus, talk about the nuclear envelope. It encloses the nucleus, separating it from the cytoplasm. What is the benefit of housing DNA in the nucleus? Why do we separate it? Bacteria don't separate it, right? Bacteria, DNA and the cytoplasm there's no boundary between the two. What's the benefit, or why do we need to separate it?
Is it because the eukaryotic cells will be more complex than the prokaryotic?
Complexity, yes. All right. So the other thing is that as humans, there was a paper I read the other day, an article said our DNA has thousands and thousands of dormant viruses inside it. Okay? Our DNA has that. So, are you infected by viruses all the time? Yes or no? Yeah. Oh, do you get sick all the time? No. Like today you're probably going to run into, and cells will be infected or exposed to, I should say, it's a better term, exposed to hundreds of viruses. Okay? Will you most likely get sick? No. Why? Let's think about this. What happens to a virus? What happens when a virus infects a cell? Here's our cell. Happy as can be. We can draw a little nucleus, a little ER. There's a mitochondria, right? And then out here, and this is just a general virus. Ugly looking thing. What's inside the virus though? Viral DNA. Okay? What's going to happen when that virus infects the cell? The DNA is going to go inside the cell. So, here we go. All right, little infection. DNA is inside the cell. DNA gets inside the cell. Where is all the DNA that is cellular DNA? Inside the nucleus. Where's the viral DNA? Outside the nucleus. So what's the advantage of having the DNA inside the nucleus? It's a protection mechanism. You can recognize viral DNA, okay, and then you have enzymes that will go and chew it up and get rid of it. So then you go and gets rid of that, and that cell doesn't become infected or doesn't produce more virus. Okay? So that is one evolution, the one way that the cells have evolved from prokaryotic cells to archaea, and then eventually to eukaryotic cells that have the nuclear envelope. Okay? So nuclear membrane is a double membrane. Each membrane consists of a lipid bilayer. All right, so you have a double layer. All right, within that, the nucleus. It's shaped by something called the nuclear lamina. Okay, and the nuclear lamina is composed of proteins such as what we call lamin A/C proteins. Okay. Now, if I told you that there's a genetic disease that exists, that this gene, this lamin A gene, what you have right here, is disrupted. Okay? If it's disrupted and it doesn't work the same as normal gene, what would you expect to happen between different cells? So if I had two cells, right, one cell here, one cell there. One is what we call wild type. It has two copies of it. And we have one that's like the knockout. Or it's this disrupted lamin gene. It has a disruptor, doesn't have the lamin gene. What do you think is going to happen? What's that cell going to look like? Knowing what the lamins do. The shape will be the shape of what? Shape of the nucleus. So now I draw the nucleus and I draw it like this. That's one. And if I had this one, it's kind of like that. Shape of the nucleus is weird, right? All right. Pretty simple, right? Pretty straightforward. But now, as a human, every one of your nuclei is disrupted in shape. What do you think happens to you when you develop or when you grow? Could that cause problems? Yeah. All right. And one of those problems, which you may have heard of, is a disease called progeria. Do you know what that is? Anyone ever heard of that before? Who said it? You age very quickly. All right. So those individuals who have progeria, progeria is actually what we call a laminopathy. You have a disruption in the lamin A gene. Your cells, right, don't divide the right way. Why don't they divide the right way? During mitosis, what happens? Cells replicate. And the nucleus has to be replicated. But now the lamin gene isn't present in the right way, so the nucleus is misshapen and the division doesn't work the same. Okay? So these individuals have this genetic deficiency or disorder throughout all their cells, and they end up what we call aging quickly. All right? So here's our nuclear envelope. Here's a scanning electron micrograph showing the nuclear envelope. And you can see these little dots. Right there. They're all over the place. It's kind of rough. You can actually see them very good over here. Okay? Those are the pores. All right, so we have this double membrane. These are pore complexes, nuclear pores, kind of all over the place, little dimples. What's important for those pores? Why have the pores? Oops, sorry. So proteins can leave the nucleus. So proteins can go into the nucleus too, right? What else has to leave the nucleus?
RNA.
RNA has to leave the nucleus. Eric.
I would say just have an overall level of homeostasis for the cell.
Yes, and that has to do with moving proteins in and out, RNA in and out. Okay? But it's a transport. Rather than putting a string of RNA, which is, a single-stranded molecule, that has to get through the nucleus, rather than having that pass through the bilayer, right? The double membrane as it is. Okay? We could just put them through the little basket hole, the pore, and have it pass through that way. That's easier and more efficient than having it pass through the double membranes. Okay? So, plenty of pores. Okay? This is what they look like. They look like these little, like I said, basket complexes. Okay? And this is a diagram of them. You will see there are ribosomes that are in there of the nuclear envelope. The double membrane that kind of loops over at the pore complex. All right? Here is important. Here's your nuclear envelope, okay, right here in purple, double membrane. And the outer membrane of the nuclear envelope is actually continuous with the rough endoplasmic reticulum. All right, so we're gonna talk about the nuclear envelope. We're gonna talk about the ER. We're gonna talk about the smooth ER. We're gonna talk about the plasma membrane. The plasma membrane, the ER, and the nuclear envelope are all part of what we call the endomembrane system of the cell. All right? Really, the plasma membrane is an extension of all of these. All right? All of this is made up of what do you think all of this is? If this is continuous with the nuclear envelope, the endoplasmic reticulum is a phospholipid bilayer. The plasma membrane is a phospholipid bilayer. The smooth ER is a phospholipid bilayer. Okay? And inside the nucleus, we have something called DNA is packaged in a form of what we call chromatin. Right? What you see there are a bunch of proteins, little circular proteins, with the blue DNA wrapped around it. Right? It's a way of packaging the 6 feet of DNA you have inside a very small nucleus. All right, and here you see the nuclear lamina, those proteins that produce some of the nuclear shape. Okay, so chromosomes, we know DNA is, how many chromosomes do humans have? 46. Okay, 46 chromosomes, 23 copies, right? 2 copies of 23 chromosomes. Chromosomes, autosomes 1 to 22, XX or XY. Okay? Each chromosome, 1 through 23, okay, is composed of a long strand of DNA. Okay? But it has to be packaged into a very small nucleus. To do that, the DNA is wrapped around in a very ordered fashion around proteins and then packaged in a form of chromatin. Right? So it condenses, okay, and so that you can fit all that DNA inside the nucleus. Right? So DNA in a normal cell, in a non-dividing cell, is found in the form of chromatin. Okay? But when the cell divides, it actually further condenses into chromosomes to separate it out. So that each of the 2 new cells have 2 copies of each chromosome. All right, within the nucleus is also a dense region, a more dense region of RNA, which is called the nucleolus. All right, and that is where ribosomal RNA is synthesized. So you have this really dense region inside the nucleus called the nucleolus. Questions on the nucleus? So, houses DNA. DNA is in a condensed form. Yes?
In the nucleolus, Are ribosomes like made there?
The ribosomes are not made there. Well, some of them can be,
Okay.
okay? But really it is the ribosomal RNA that's synthesized there, that's the big part,
Okay.
okay? Other questions on the nucleus? All right, the next part of eukaryotic cells, and this is important because all cells have these, are ribosomes, okay? Ribosomes are complexes of ribosomal RNA and protein, all right? They are what we call a tri-complex. They have two protein subunits, okay? A large subunit, we call an 18S, and a smaller subunit, we call a 16S. And I call it 18S, 16S, because when they were discovered, they were based upon S is for sedimentary units. It has to do with fractioning the cell and then sedimenting the proteins out on a sucrose gradient, blah, 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 blah. You don't have to know it, but just know that one is bigger than the other because it sinks further than the other, okay? So, and in between these two subunits is a little bit of ribosomal RNA that allows them to stick together, okay? That is the structure of ribosomes. All right, we'll get into more of the structure of ribosomes and how they function either very late this semester or early next semester. All right, the role of a ribosome is to carry out protein synthesis. Okay, and this happens in two separate locations. All right, it happens where? Where are ribosomes found? In the cytosol and on the rough ER. All right, so those two locations is where protein synthesis is done. Okay? Why is it important why do you think it may be important for ribosomes to be located on the rough endoplasmic reticulum? Think about it. Talk to your neighbor about it. Come up with something. Okay? Come up with something. Here's some hints. Think about what ribosomes do and where they're located.
Maybe I'm wrong.
That's just the two sub.
The only change was stuff. Chain is threaded into element.
You took notes, huh?
We took notes.
All right, what do we got?
We said that might be because like DNA or RNA leaving the nucleus would like get caught by the ribosomes and like form proteins that they need to like form.
Yes, this is true. Absolutely. So RNA leaving the nucleus will be bound to ribosomes, right? Either in the cytosol or on the rough ER. What is the rough ER? What is it? It's a bilayer. Okay? What are proteins going to produce? Or what are ribosomes going to produce? Proteins. Okay. What else is made up of a bilayer? Just, all right, we're gonna do popcorn here. Just come out what you want. Just yell out where you're coming, what comes to mind. What else is produced or composed of a bilayer? The nucleus, the nuclear membrane, what else? The cell membrane, right? All the other things that are inside a eukaryotic cell that have membranes or bilayer right around them. All the other organelles, right? Okay. So here we have this endoplasmic reticulum with ribosomes on it, right? What else is the plasma membrane, for instance, composed of? Not just a bilayer, but what else? Proteins. All right. So now here in the ER, in the rough ER, we have a ribosome that's already embedded or attached to a bilayer. Now it's going to produce a protein, right? That may be embedded or already embedded inside the membrane, that it can go and produce it on either side. Is that an easier way to produce a membrane protein, right? A protein that's in the plasma membrane, inside the membrane, rather than producing the protein in its structure, three-dimensional structure, and then having to put it inside a plasma membrane. Because what do we know about the plasma membrane or a phospholipid bilayer? Part of it's hydrophobic, part of it is hydrophilic. What are the chances that that folded protein is going to be hydrophilic or hydrophobic? Or be easy to get embedded inside that that bilayer? Is it going to be easy to do? No. Okay, so membrane proteins in the membrane, in the plasma membrane, in the rough ER, in the nucleus, in the nuclear membrane, they are produced here. They're not produced in the cytosolic protein, cytosolic ribosomes. Why? Because it allows the cell to put the membrane or the protein directly into the membrane, right? And so you can put it in as an amino acid sequence in primary structure before it gets to the tertiary structure where it won't fit. All right? That's a huge advantage. Is it much more efficient, right? Rather than having a protein that has to be unfolded, right? Embedded into the membrane and then refolded in its right structure. Okay? So that's a huge advantage to having those ribosomes bound on the ER. All right? So this is what it looks like. Okay, here's an electron micrograph showing. So you'll see that there's probably 1, 2, 3, 4, 5. There's a bunch of ribosomes on this bilayer right there, right? And then there's a bunch of ribosomes in between them. Right? So you have both free and endoplasmic reticular or bound ribosomes on the ER. Okay? And there's your diagram of your large versus small subunit ribosomes. So, let me ask you another question. How many domains are there of life, of cells? 3 domains, and those domains are what? Bacteria, archaea, and eukarya. Okay. Do all 3 of those domains of cells have ribosomes? Yes. Okay. So we have 3 domains. They're all a little different from each other, but they all have ribosomes. So what can you compare in those cells to show that those bacteria are different from archaea, are different from eukarya? What could you compare? Different kinds of RNA. Okay. What if not all cells have RNA? What proteins? Okay, let's go back. We'll do it again. What do all the cells have? Ribosomes. Okay. So what can we compare to show that all those cells are a little different? Ribosomes. Because would you expect that cells have these things in common, right? That if they are supposed to be different, and they're going to live in different areas and do different things, if they're going to be different, that the ribosomes would be a little different. Yes? You agree? All right, good. So that is what researchers did to show, years ago, to show that bacteria ribosomes are different from eukaryotic ribosomes, okay? And now, what's the other domain that we're talking about? Archaea. So, comparing ribosomes from archaea versus bacteria, right? Those two were very different, all right? Then comparing ribosomes from archaea to eukaryotic cells show that the ribosomes were less different. They were more alike than from archaea to eukarya than archaea was to bacteria. Right? And what did I show you before? It was kind of like the tree. We had bacteria, right, over here. And then over here we had, all the way over here, we had eukarya. You remember this? Where was archaea? Off of the eukarya. Okay? Why? Well, one piece of evidence for that was comparing the ribosomes. Okay? So it turns out that the subunits of bacteria ribosomes are different sizes than archaea and eukarya ribosomes. Okay? So thinking about the information that we have, all cells have ribosomes. Not all cells are a little different. Well, let's compare the differences. All right, and let's see how they are different. All right, questions on ribosomes. Their main function, protein synthesis. All right, and I just kind of gave a clue a little bit ago. I mentioned the endomembrane system. Okay, the endomembrane system of a cell includes the nuclear envelope, the ER, the Golgi, the lysosomes, the vacuoles, and the plasma membrane. These are all things that are composed of what? A phospholipid bilayer. Okay? So phospholipid bilayer, because they are composed of that, it allows them to fuse with one another. All right? Cells make proteins. Yes or no? Yes. Sometimes cells make proteins that they need to get out of the body or get out of the cell, what we call secrete proteins. Okay? How do they do that? How does a cell produce proteins inside the cell and then eventually let it go? This happens all the time. It's happening in your eyes right now. It's happening in your ears, in your nerve impulses that are being done. Okay? Every time there's a nerve impulse, at the end of an axon, there are little pieces of proteins that are held in axon vesicles, okay, that they store. And then those axon vesicles fuse and they release proteins out to stimulate the next axon or the muscle or whatever else, right? It's a release of what we call a neurotransmitter. Hey, those are proteins that have been produced by that nervous cell, that neuron. Okay? They've been held inside the little vesicle until they fuse and they release. Well, how does a cell do that? That is all a function of the endomembrane system. Okay? Because at the end of an axon, it kind of looks like this, here's a little vesicle. Both of the vesicle and the axon cell membrane is composed of what? Phospholipid bilayer. So when bilayers come together, what's going to happen? You ever take a bubble solution when you're a kid, you blow bubbles, right? right? You blow bubbles. My daughter does it all the time. Blow bubbles. You get two bubbles that come together. What happens when the soap comes together? Eventually fuses into, it fuses into a bigger bubble. Why? What is that bubble solution? What's going to happen here? What's going to happen here?
It's the same.
It's the same thing. This is a bilayer. That's a bilayer. It has hydrophobic and hydrophilic regions. So when it comes together, right, it's going to go and it's going to fuse with it. You're going to end up something like this now. Right? And then eventually whatever proteins were inside there are released into the synaptic cleft. Okay? So because of all these things, or the nuclear envelope, the ER, the Golgi, the lysosomes are all composed of a phospholipid bilayer, it allows you to move the cell to move things around and export it. All right? Or bring it in. Okay? Or bring it in. All right? So all of these things, they're either continuous or connected through transfer by these little membrane blebs called vesicles. Okay? So the ER. The ER is your biosynthetic factory. What types of ER are there? Rough and smooth. What's going to be produced? Because what's biosynthetic means what? Biosynthetic. Gina. You said something.
It like, no, I don't know.
Made in the body, made in the cell, made for life. Synthesis, we're going to produce something, right? Bio, life, right? So this is your biosynthetic factory. What's going to be produced or made in the rough ER? Proteins. Why proteins?
Because of the ribosomes.
Because we have ribosomes, all right? So proteins are going to be there. In the smooth ER, what do you think is going to be made? Lipids. Why? Because it's composed of phospholipids. All right? So the production, we have a bunch of enzymes that are present in the smooth ER that are going to be important for producing lipids. Okay? The rough ER is going to produce membrane-bound proteins and other proteins. The smooth ER is also going to work for what we call detoxification. Right? It's going to break down some of the stuff that the cell it's not good for the cell. All right? And that happens. Some of these, the rough ER and the smooth ER, can change depending upon the cell type we're talking about. Okay? It can have different functions depending upon the cell type. All right? So here's the nuclear envelope, the rough and smooth ER. Okay? Your book mentions this little thing called the transitional ER. What do you think that is? It could be both, or it's the area between one and the other. Okay, a connection. All right, but a little bit of a difference in structure between the smooth ER and the rough ER. Look at how convoluted this bilayer is as opposed to this. Why? Different structure, different function. What's giving it the different structure, though? The studded ribosomes. So those ribosomes are pretty equally separated from one another, right? Yeah? That actually produces, or because of their structure, or how equal they are along their separation, that keeps the ER in a definitive structure. All right? With less or no ribosomes, you end up with a more convoluted structure of the lipid bilayer. Okay, here's your smooth ER, some functions. It synthesizes lipids and metabolizes carbohydrates. What does that mean? Right? It helps break things down and will help metabolize, break down carbohydrates from larger polysaccharides to smaller monosaccharides. So then glucose can be packaged into or sent to the glycolytic cycle, glycolysis, broken down, and then lead to cellular energy. All right, but that's the first step. Detoxifies drugs and poisons, and then here's another one. In certain cell types, it stores calcium ions. Okay? Anyone know what cell type that would be? Muscles. Okay? In muscle cells, it stores calcium ions. And in muscle cells, the smooth ER or the endoplasmic reticulum is called a different name. It's called the sarcoplasmic reticulum. Because it's sarcoplasm refers to muscle, all right? Or sarco is a prefix that actually refers to flesh, but muscle, all right? So sarcoplasmic reticulum in muscle cells. Questions on the ER? The rough ER. Here you go. It has bound ribosomes which secrete what we call glycoproteins. All right? So really, the rough ER is going to produce proteins. Okay? Whether or not they're glycoproteins is irrelevant. All right? But glycoproteins are proteins that are covalently or linked to a carbohydrate. We're going to actually put a small piece of carbohydrate onto an area of the protein. That's all that is. All right? So we're going to talk about glycoproteins and what they are. We could have glycoproteins, you could have lipid-linked proteins. And basically, it's a protein that has a piece of carbon or carbohydrate portion to it. All right? Within that, it's going to produce those proteins, and then it's going to produce what's called transport vesicles, proteins surrounded by membranes. So we need to get some type of protein, insulin, right? We all know what insulin is, right? I've heard of that before. Where's insulin found? It's produced in the pancreas. Where does it work? In blood, right? And then it actually goes, it's transported in the blood and it goes to all cells, okay? So if I told you insulin was a protein and it's produced in the beta cells of the pancreas, where in those beta cells do you think it's produced? The rough endoplasmic reticulum. Why? It's going to be secreted from the cell, okay? And this is, so we'll use insulin as our example. In the beta cell, it's produced and produced into a transport vesicle, okay? And then from there, in the rough ER, it's going to go from the rough ER to the Golgi apparatus. And Golgi apparatus is like it's like the mailroom, okay? It's your shipping and receiving, okay? You get something made from manufacturing. Now, I don't know where this is going, but here I made it. You take care of it now. So now the Golgi says, all right, well, this one's been ordered. We know where it's coming from. We know where it's going. We can tell, and we ship it off to where it needs to go. So it gets packaged and modified and then shipped off. Yes, sir?
So it says it's a membrane factory, so it also makes like the bilayer too?
It actually makes the bilayer too. And that goes back to what we talked about before. If we produce a protein that's inside the bilayer, it's already there, right? And then that portion of the lipid bilayer can be moved and go as part of the vesicle to the membrane. Okay? So it kind of looks like this. If we had, here's our ER, right? No. Let's erase that. Here's our ER, and here's our plasma membrane. Okay? So we'll just do PM and ER here. Do the rough ER, we'll stud some ribosomes on there. All right? Now, protein, say we need a protein that has to be reproduced in the plasma membrane. All right? So this ER is going to produce it, and it's going to actually be produced inside a vesicle. Okay? As part of the membrane. That vesicle, then it's going to go to the Golgi, and then be transported out. But it's still embedded inside that vesicle, right? Now this vesicle is going to fuse with this plasma membrane, right? So now it fuses, and now that protein is part of the membrane. What it ends up becoming, it ends up looking like this. Now you have a protein embedded inside the membrane, and it's already there, okay? Instead of producing a protein, having it fold all funky, and then having to refold it and embed it, just produce the membrane chunk, and then that gets packaged and transported to the plasma membrane itself. So yes, it is it is a membrane factory. The Golgi apparatus, I just said, is a shipping and receiving center. Okay, it's a FedEx location. So it consists of flattened membranous sacks called cisternae, right? It is still a bilayer. What's it going to do? It's going to modify the products of the ER. ER is going to produce that proteins. It's going to modify them. Sometimes it will add carbohydrates. Sometimes it will change it and make sure it's folded the right way. Whatever it does. It's like quality control inside and shipping and receiving. All right. The Golgi will manufacture certain macromolecules, but that is a very small, insignificant function that it does. All right. But really, the main part is this. It sorts and packages materials into the transport vesicles, which then will be pushed out of the cell or to the plasma membrane, wherever it might be. Yes, sir.
So is everything that's made in the cell goes to the Golgi apparatus to get sent out?
Pretty much.
Okay.
Yes. Because there has to be an address label. Okay? A cell's gonna produce whatever's there, okay? And then it's gonna determine where it's going. Okay? Using proteins as an example, the two places where proteins can be made is in the cytoplasm on cytosolic ribosomes, and in the rough ER on membrane-bound ribosomes. Okay? 90% or 95% of cytosolic proteins that are going to be found in the cytosol, are made on cytosolic ribosomes. Okay? That's where they're going to be. We don't have to move them around, they're not going anywhere. All other proteins, 99.9% of the proteins that are produced that are going somewhere, have to be in a membrane, have to go to the nucleus, they have a destination, they have an address label as part of the protein itself. The Golgi reads that address label and tells it where to go. We'll package it and send it on its FedEx truck, its transport vesicle, to where it needs to go. So if a protein is, part of it says, that's a membrane-bound protein, put it into a vesicle, set it to the plasma membrane. If it needs to go to the nucleus, put it in a vesicle, send it back to the nucleus. If it has to go back to the rough ER, put it in, send it back. Okay? Everything moves through the Golgi. Okay? And this is what it looks like. There's two-sided, okay? There's a receiving side and a transport or shipping side. The receiving side takes things from the rough ER, okay? So vesicle coming from the rough ER is going to come and fuse with these vesicles, and then there's going to be enzymes and stuff in here that's going to modify it, blah, 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 comes through, and now we know where it's going. The shipping side is now going to produce another vesicle and send it off to where it needs to go. Here's your receiving side, here's your shipping side, there's a transport vesicle with material X inside it. Okay? Everything is transport that the cell's going to transport in or out is going to go through the Golgi. Okay, we'll stop there for today. Questions? Okay. Other thing, you will have a quiz on Friday. Okay, quicker quiz on Friday on chapter 4. I got through 15 slides.